Thank you very much, Trudy. My name is Dr. Andrew Thompson. I am a neuroscientist working with the Research and Development Group at Sovereign Health. And today I'm going to be providing sort of a primer introduction to the brain, focusing largely on the physiology and pharmacology of the brain. So as this is an introductory level course and designed to be sort of a primer, there's going to be a lot of terminology that I go over. So let's start by introducing some terms. First of all, we need to define physiology and pharmacology. Physiology is defined as a branch of biology that deals with the normal functions of living things and their parts. Physiologists tend to divide the body up into systems that work together. So for instance, you might have the digestive system, the cardiovascular system, the nervous system, which is going to be the focus of our talk today, the muscular system, and the skeletal system. And these systems all work together to create a functioning organism. Pharmacology is a branch of medicine that is concerned with the uses, effects, and modes of action of chemicals that affect the body. And these chemicals can range from drugs of abuse to therapeutic drugs to some toxins. There is a wide variety of chemicals that have various effects on the body, and pharmacology is the study of these. The main thing that we're going to be talking about today is the brain. So we need to introduce the brain and learn a few things about it at first. The brain is a part of the nervous system, and it serves as a control center for the rest of the body. It makes up about 2% of the body's weight, but it uses up about 20 to 25% of all the energy that's produced by the body, so it's a vitally important and energy-hungry organ. It's made up of approximately 86 billion cells called neurons and approximately 85 billion cells that support those neurons, which are known as glia. So in order to familiarize ourselves with the brain before getting into an in-depth discussion of the pharmacology and physiology of it, I'm going to take us on a brief tour of the structure of the brain. The study of the structure of the brain and the nervous system is called neuroanatomy. And I'm going to be using a tool as I take us on this tour called 3D Brain. It's a project by Genes to Cognition from the Cold Spring Harbor Labs. It's available in the App Store or online for free at www.g2conline.org, and it's a helpful tool for helping yourself learn neuroanatomy if you're interested after this webinar. So, and most of the images that I show are going to be from that website. So this brain can be subdivided into 5 major subdivisions. The one that we most often think of when we think of the brain is this outer surface of the brain, which is called the telencephalon. And deep inside the telencephalon is a structure called the brain stem, which you can see here zoomed out. So at the top of the brain stem, inside the brain, is a region called the diencephalon. And then working our way backwards from there, we have the mesencephalon, the metencephalon, and the myelencephalon. And so these terms are going to be important just as landmarks for finding our place within the brain. As we go, I'm actually going to take us on a tour starting from the myelencephalon and moving our way up to the telencephalon. So the myelencephalon is in the back of the brain stem at the bottom, and its main structure is the medulla, which controls many essential brain functions that are necessary for life, including breathing and heart rate. It also serves as a way station for any ascending tactile information from the skin. So if you feel something touch your skin or you feel heat, then that information is carried up and stops in the medulla before traveling on to its destination elsewhere in the brain. Moving up, we have the metencephalon, which consists of 2 major regions, the pons and the cerebellum. Pons is Latin for bridge, and this is actually what it serves as, a bridge for signals entering into the cerebellum. It also has a role in monitoring and adjusting breathing rate. The cerebellum sits at the back of the brain. It's this purple structure that you can see in the image on the bottom left, and it's responsible for refining motor control, coordinating motion and balance, and the way that it does this is that it actually gets a copy of all the information of what the brain wants the body to do, and it also gets a copy of the information coming up from the body of what the body is actually doing. And it compares these 2 sources of information and detects and corrects any mismatches. So it's a very important part of the brain in order for fine motor movements to be well coordinated. The next stop on our tour of the brain is the mesencephalon, also known as the midbrain. And this section has two major regions, the tectum, which is Latin for roof and is sort of on the top of the midbrain, and the tegmentum, which is Latin for floor and is on the bottom of the midbrain. The tectum serves as a way station for incoming visual and auditory information, and it's useful for helping to localize the sources of sounds that you hear in the environment and moving your eyes towards visual stimuli that appear, maybe in the periphery of your eyes. So if you notice something off in the distance and your eyes move towards it, you can thank your tectum for that. The tegmentum is responsible for carrying motor commands from the higher regions of the brain down to the spinal cord where they go out to the muscles to be enacted, and to the pons where they go over into the cerebellum to be compared against information about what the body is actually doing. The tegmentum is also very important for the prediction and detection of reward and for the initiation of movement. The next subdivision of the brain that I'm going to talk about is the diencephalon, which consists of the thalamus, which is a region deep inside the brain that mainly serves as a relay station for any sensory information. So any sensory information coming from organs like the eyes, the skin, or the ears makes a stop in the thalamus, and then it's routed off to its proper destination elsewhere in the brain. The one exception to that is the sense of smell, which actually bypasses the thalamus for reasons that are not quite clear. The other part of the brain that is in the diencephalon of note is the hypothalamus, which is located just below the thalamus, hypo being Latin for below. And the hypothalamus is responsible for controlling all the circulating levels of hormones in the body and the blood. And this means that it regulates a lot of behaviors, and the main behaviors that it's involved in are what we call the 4 F behaviors, which are feeding, fighting, fear, and mating. Moving up to the telencephalon, this is, again, like I said, the part of the brain that most people think of when they think of the brain. And it's made up of a lot of regions, which I'm going to show you in a moment. The basal ganglia is responsible for reward and reinforcement and movement initiation, and has a role in learning how to do things, or skill learning. The hippocampus is involved in memory formation and spatial learning, or learning how to construct mental maps of your environment and learning where things are located. The amygdala is responsible for emotional awareness and for learning about things that might be fear-inducing. And the corpus callosum is actually slightly different. It's a structure that connects the cortices of the left and the right hemispheres of the brain. And the last part of the telencephalon that I'm going to talk about is, of course, the cerebral cortex. So let's look at these subcortical structures first. Subcortical structures meaning deep within the brain below the cortex. The caudate, this C-shaped structure in purple on the left side of the image here, and the putamen, the sort of pancake-shaped structure, and I guess that's a mild pink, make up the basal ganglia along with some other local regions. The amygdala, shown here, is a little almond-shaped structure that's visible in both images, and it sits just above the hippocampus, which is shown here in green. The corpus callosum is a bundle of neural fibers that crosses from one half of the brain to the other, crosses the midline there, as you can see in the left image. And it carries information from the cortex of the left hemisphere to the cortex of the right hemisphere and back, and it's responsible for allowing your brain to work as a unified whole rather than two half brains, which it would otherwise. Finally, we reach the cortex, which is, again, the most familiar part of the brain. You can see it has a very distinctive structure with some bumps and grooves. The bumps that you see are called gyri, or singular gyrus. The valleys between the bumps, or the grooves between the bumps, are called sulci, or singular sulcus. And these sulci provide us with some important landmarks. So the first one I'm going to mention is the longitudinal fissure, which is, a fissure is just a name for a big important sulcus. The longitudinal fissure goes directly between the two hemispheres of the brain and divides them in half. And it separates the left from the right. Again, that goes down to the level of the corpus callosum, and the corpus callosum actually crosses over the longitudinal fissure. The other important landmarks that we want to talk about are the central sulcus, visible in the image on the left, which runs up and down from the top to the bottom of the brain, and it actually divides the frontal lobe from the lobe that's just behind it, or the parietal lobe. The third landmark is the lateral sulcus, which is on the side of the brain. It's sometimes also known as the Sylvian fissure, and that divides the frontal and parietal lobes from the temporal lobe on the side. And the last major landmark on the brain actually isn't visible from the exterior surface, but if we sort of make half of the brain clear as we've done on the right, you can see there's a sulcus visible on the internal surface of the brain that separates the parietal lobe from the occipital lobe, and that is the parieto-occipital sulcus. So I've been talking about these lobes, but let me just get into a little more detail about what they are. The frontal lobe is located at the front of the brain, and it's responsible for a lot of planning and executive function. The parietal lobe is located just behind that, just divided from it by the central sulcus, and it's responsible for spatial relations and making associations between things. The temporal lobe is located on the side of the brain by the temple, and it's responsible for hearing and memory and some speech production. And the occipital lobe in the back of the brain is responsible for vision. So with that, we've completed our quick and dirty tour of the brain, and we can move on to learning a little bit about the physiology and pharmacology of the nervous system. So I'm going to start with neurophysiology, which is the study of the functioning of the nervous system. As I said, physiology is the study of the functioning of various systems. Neurophysiology, therefore, is the study of the functioning of the nervous system. This can range in scale from the molecular functioning to how the cells of the nervous system work to how the whole nervous system works together as a whole. For today's purposes, I'm primarily going to focus on mechanisms of cellular communication as they relate to neurophysiology. The cell in question here is the neuron. So this is a schematic sort of cartoon image of a neuron, but it shows many of the important features. So just like every cell in the body, a neuron has a cell body. And this is where the nucleus of the cell is. This is where a lot of the cellular machinery takes place that keeps the cell alive. Branching off of the cell body, you can see there's a bunch of, we call them processes. They are bits of cell that branch off of the main cell body and do things. The most numerous of these are called dendrites, and dendrites serve as the input region to a neuron. And each neuron can have as many dendrites as it needs, but each neuron only has one axon. So the axon is a cellular process that goes out from the cell body, and it's actually going to be the output region, and it will carry information in a way that we'll get to in a moment. At the end of the axon, there's another branching region, and this is called the axon terminal, and this is going to be the output region of the neuron. So you can see that the axon terminal sits in very close proximity to the dendrites of another neuron. And in fact, they don't touch, they just get very close to one another and form what is called a synapse. So these synapses are the sites of cellular communication in the nervous system. These are how one neuron talks to another. The other part of the neuron that I want to call attention to is this little region here at the beginning of the axon, which is called the axon hillock. And that's going to become important in a moment. So this is sort of a cartoonish schema of a neuron. What do neurons actually look like? Well, we know that thanks to, in large part, to the work of a scientist named Santiago Ramon y Cajal. And he sketched neurons that he visualized using a technique called the Golgi method. And the Golgi method causes neurons to turn entirely black and lets you see all of their processes and lets you see the axons, here indicated with a lowercase letter A in Cajal's drawings, and they let you see the dendrites and the cell body. However, there's a catch, which is that this method only stains about 1% of all the neurons. That actually helped out Dr. Ramon y Cajal, because he was able to see individual cells in high levels of detail and visualize their structure and recreate that structure in art. In the actual brain, what that looks like is something like this. And so these are Golgi-stained neurons. On the top, we have a sample of the cortex. On the bottom, we have a sample of the hippocampus, and these are actually from a mouse. These are Golgi-stained neurons, and remember how I said that it stains about 1%? Well, you can see that there's a lot of empty space in both of these images. So this gives you a great appreciation for the complex structure of neurons, but it doesn't necessarily give us an appreciation for how densely these neurons are packed in the brain. So for that, I'm going to turn to another technique, which is known as brainbow. And what this does is it causes every neuron in the brain to turn a different color. And this is what we see. So up on the top picture, again, is a sample of mouse cortex. And the bottom picture is a sample of mouse hippocampus. And you can see how tightly packed in these neurons are and how many of them there are. Remember, the human brain contains about 86 billion of these neurons. So now that we've become familiar with the structure of a neuron, we can start talking about the function. And in order to talk about how neurons communicate, I'm going to introduce some new terminology. Each neuron is surrounded by a cell membrane, which is a dual-layered, semi-permeable barrier between the inside of the cell and the outside of the cell. And actually, every cell in the body is surrounded by one of these cell membranes, but the cell membranes around neurons are special. And the reason that they're special is because they have what's called membrane potential. The membrane potential is a difference in voltage between the inside of a neuron and the outside of the neuron, or the extracellular fluid. And this potential, or difference in voltage, is going to be very important for understanding how neurons communicate with one another. So where does it come from? Well, on either side of the cell membrane, there are ions, which are charged particles, and they are unevenly distributed between the intracellular and the extracellular fluid. So if you were to, as shown in this cartoon, place a reference electrode outside of the cell and a recording electrode inside the cell, you would detect that the inside of the cell is about 70 millivolts less in voltage than the extracellular fluid. So we call this the resting membrane potential of a neuron. And this is the case when the neuron is not active or doing anything. So in this state, since there's a difference between the internal and the external voltage, we call it polarized. So since it's at negative 70 millivolts compared to the outside, the neuron is considered polarized. If this difference in voltage between the inside and the outside were to shrink, in other words, if it were to become closer to 0 millivolts, or were to invert, in other words, go greater than 0, making the inside of the cell positive relative to the outside, then we refer to the neuron as depolarized. Conversely, if the difference between the inside and the outside of the neuron increases or gets further from 0 millivolts, the neuron is hyperpolarized. And these terms are going to be very important when I start talking about neurocommunication between neurons. So right now we're just thinking about within one neuron how information is passed. So these ions are capable of moving across the membrane, and their movement, or their tendency for movement, is determined by the driving force on the ion. The driving force is a result of the chemical and electrical properties of the neuron. And what I mean by this is that molecules will tend to travel from areas of high concentration to areas of low concentration. So if you have a lot of one molecule in one area and not a lot of that same molecule in another area, you've set up a chemical gradient, and the molecule will tend to move from the area of high concentration to the area of low concentration. This is called diffusion down a chemical gradient. Conversely, in addition, charged molecules, like ions, are attracted to molecules or regions with the opposite charge. So molecules carrying a positive charge are going to want to go towards a negative charge, and molecules with a negative charge are going to want to go towards a positive charge and away from regions with the same charge as them. And this is referred to as electromotive force. So the electromotive force and these chemical gradients on each ion combine to determine the driving force on the ion. And again, the driving force is the tendency that a ion would want to move, the direction that the ion would want to move if it were able. So at this point, at rest, the ions are unable to move because the membrane is not permeable. So the major ions in question here that are involved in the potential are sodium, potassium, chloride, calcium, and some other molecules that sit inside the cell and don't do very much. Sodium, which has the symbol Na+, indicating that it has a positive charge, has a very high extracellular concentration and a relatively low intracellular concentration. So the chemical gradient on sodium favors the movement into the cell. Additionally, since the inside of the cell is negative relative to the outside, sodium wants to move in based on its electromotive force. So when you combine these together, the driving force on sodium, the direction that it would move if it were able, is inward. Potassium, conversely, has a low concentration outside of the cell and a high concentration inside of the cell. Therefore, the chemical gradient direction is outward. Potassium wants to move out of the cell from an area of high concentration to an area of low concentration. However, potassium, whose symbol is K+, indicating that it has a positive charge, wants to move toward a negative charge, and remember that the inside of the cell is negative relative to the outside. So potassium, based on its electromotive force, wants to stay in the cell. This results in a relatively balanced state for potassium, where potassium doesn't really move in or out of the cell that much. Chloride is in a similar situation. Its symbol is Cl-. It carries a negative charge. There's a lot of it outside of the cell, not a lot of it inside the cell, so it wants to move in based on the chemical force. However, since it is negative, it wants to move out based on the electromotive force, so it ends up being relatively balanced. Calcium is very similar to sodium in that outside of the cell, but a much lower concentration inside the cell. So the chemical and electromotive forces push it inward. If it were able to move. These other molecules are things like DNA, folded proteins, which carry a negative charge and are highly concentrated inside the cell. There is a lot of driving force on these ions to move out of the cell. However, the membrane is not permeable to these cells. Therefore, they can't move. So when I've alluded to the next topic, which is permeability, and the cell membrane is, as I mentioned, semi-permeable, meaning that it is sometimes permeable to some things and sometimes permeable to other things and sometimes not permeable at all. And what makes that determination are molecules called ion channels. And the membrane contains a lot of these. These are specialized holes which permit certain ions to pass through the membrane under specific conditions. And there are a bunch of ion channels, but for our purposes right now, there are three that we're really going to be paying attention to. And these are the voltage-gated sodium channels, which are located at the axon hillock and all along the axon. And these open if the nearby membrane potential reaches approximately negative 55 millivolts. So if the cell depolarizes a little bit from minus 70 to minus 55, these sodium channels are going to open and the membrane is going to be permeable to sodium. There are also voltage-gated potassium channels located all along the axon and voltage-gated calcium channels, which are located specifically at the axon terminal. And these are all going to be important elements of neural conduction and communication. So when a depolarizing stimulus or an electric charge is detected at the axon hillock, it changes the membrane potential. It makes it slightly more positive. If this stimulus happens to be strong enough to elevate the membrane potential to the threshold for the sodium channels to open, the voltage-gated sodium channels, then these sodium channels will open. This will result in sodium moving in the direction of its driving force, which is very strong inward, and sodium will enter the axon and increase the membrane potential on the inside relative to the outside. This rapid increase is the beginning of an action potential, which is the fundamental unit of neural communication. An action potential looks like this graph on the right, showing time in milliseconds on the horizontal axis and the vertical axis showing voltage. This is the voltage across the cell. So you can see that at rest it's at about -70. And the action potential consists of 5 phases. First is the initiation phase, then the depolarizing or rising phase, then the repolarizing or falling phase, then there's a hyperpolarized state, and then a return to the resting state. And these phases are all characterized by the opening and closing of certain ion channels. So we're going to walk through these phases now. Action potential initiation can occur, as I said, when the membrane potential reaches the threshold for the voltage-gated sodium channels to open. And once this occurs, sodium ions move freely into the cell in the direction of their driving force. At the same time, voltage-gated potassium channels start to open, but they open much more slowly. So as the sodium is rushing in, the cell's membrane potential is getting more and more positive, and this is increasing the voltage inside the cell relative to the extracellular fluid, and it reaches a peak of approximately positive 35 millivolts. And this is the depolarizing or rising phase of the action potential. At this point, we need to stop and reconsider what's happened to the driving forces, because now there's a lot of sodium inside the cell. There's a lot of potassium still inside the cell because the potassium channels haven't opened yet. And that means that there's a buildup of positive charges inside the cell that's trying to drive sodium and potassium outward. So now the driving force on both sodium and potassium is strong outward. Sodium channels inactivate shortly after they open, so that stops the influx of sodium, and it prevents any sodium from moving outward now that the driving force is outward. At the same time, potassium channels that originally started to open when the threshold was reached during the initiation phase have now been fully opened, and there's a strong driving force on potassium to leave the neuron. And so potassium is now free to move according to its driving force. It leaves the cell, causing the internal membrane potential to drop again, and the cell to repolarize. These potassium channels remain open a little longer than it takes to repolarize the cell, so the membrane potential actually overshoots the resting potential, and the neuron enters a hyperpolarized state, which makes it very difficult to initiate a new action potential. This is what we call the refractory period, and it refers to the inability of the neuron to induce a new action potential during this time. Of course, now there's a lot of potassium outside the cell and a lot of potassium inside the cell. So in order to restore the resting potential, the cell has to actually expend energy. And it uses a molecule called the sodium-potassium pump, which pushes sodium out of the cell and brings potassium into the cell. And this restores the normal distribution of ions that existed prior to the action potential, and it restores the resting potential so that the cell is ready to fire another action potential. So that describes the increasing and decreasing phases of the action potential. But in order to understand how it travels along the axon, we need to consider what happens to the sodium that actually entered the cell during the action potential. So sodium entering the cell during the action potential produces a region of very high concentration of sodium right at the site of entry. And this sodium has to diffuse away from there, and it diffuses actually in all directions. So some of it gets restored via the sodium-potassium pump, some of it goes up the axon, and some of it goes down the axon. Now, of course, sodium moving elsewhere in the axon is going to depolarize the next section or the adjacent sections of membrane and trigger another action potential to occur in that section of membrane. So that's exactly what happens. However, the sodium that's traveling backwards is unable to trigger a new action potential because that segment of membrane is still in the repolarizing phase, or in the after action potential hyperpolarization or the refractory period. So this is what causes the action potential to move in one direction down the axon. Notably, if you were to inject a current or inject a stimulus in the middle of an axon, there would be no reason for it to only travel in one direction, and so the action potential would actually move in both directions. But since all action potentials are naturally generated at the axon hillock, action potentials propagate down the axon in only one direction, and this is due to the refractory period. Now, I mentioned that the action potential is critical and it is the fundamental unit of the nervous system. So what happens when it doesn't work or when it breaks down? We actually do know exactly what happens because there are toxins that interact with that can prevent the action potential from working properly. One example of this is the Japanese pufferfish, or fugu. Japanese pufferfish is considered a delicacy in Japan, but it can be dangerous to eat if it is prepared by an inexperienced chef. Chefs who want to serve fugu have to undergo 3 years of rigorous training and be licensed. And the reason for this is that it contains a toxin. Part of the appeal of eating the fish is this toxin because diners experience a tingling sensation or a numbness in the mouth, and they sometimes report feeling lightly intoxicated. So that's part of the appeal of this dish. What this fish contains is high levels of tetrodotoxin, which is a molecule that sits in sodium channels and prevents them from working, especially these voltage-gated sodium channels that are necessary for the propagation of action potentials. So with these blocked and action potentials unable to fire, eating and being poisoned by fugu involves paralysis and eventually suffocation, which can be fatal. So action potentials are critically important, and disruption of them can be fatal. The next phase of action of cellular communication occurs actually once the action potential has propagated all the way down the axon and is at the axon terminal. And this is going to be the aspect of neural communication that we call neural transmission. So neural transmission deals with how the signal gets from one from one axon to the dendrites of the next cell. Again, reintroducing this terminology, the synapse is the junction between two neurons where communication occurs, and the neurons here are called the presynaptic neuron and the postsynaptic neuron. The synaptic cleft is actually the microscopic gap between the presynaptic and postsynaptic neurons. And a neurotransmitter is a chemical messenger that is released by a neuron in response to an action potential. It travels across the synaptic cleft and it binds to a receptor on another neuron to induce an effect. How does this work? Once an action potential arrives at the axon terminal of the presynaptic neuron, voltage-gated calcium channels are opened. You remember I mentioned these previously. And these work in exactly the same way as the voltage-sensitive sodium channels, in that they are opened by an increase in membrane potential. They allow for calcium to come into the cell. These are specific for calcium, though, and they don't allow sodium to come into the cell. Once calcium enters the cell, it triggers the fusion of these little packets of neurotransmitter, as you can see in this diagram on the right. These little packages of neurotransmitter are called synaptic vesicles, and they fuse when in response to calcium. They fuse with the cell membrane, allowing neurotransmitters to be released into the synaptic cleft. Now these neurotransmitters travel across the synaptic cleft from the presynaptic to the postsynaptic neuron, and once they're there, they bind to receptors. These receptors are specialized proteins which bind to neurotransmitters and induce an effect. There are many different types of receptors, but they can be broadly grouped into two families. These are the ionotropic receptors and the metabotropic receptors. Ionotropic receptors are also known as ligand-gated ion channels, and metabotropic receptors are G-protein coupled receptors, which I'll get into in a moment. First, the ionotropic receptors. Like I said, these are ion channels, just like the voltage-sensitive sodium and potassium and calcium channels, except instead of opening in response to a change in membrane potential, these channels open when bound to a ligand. A ligand is just a molecule that can bind to a receptor. In this case, we're talking about a neurotransmitter, but it could also be a drug. Once a ligand binds to these receptors, they open, allowing the ion that they are specific for to pass through. So for instance, sodium can enter the cell via an ionotropic receptor, and when this happens, again, an influx of sodium produces increase in membrane potential, and we call this an excitatory postsynaptic potential, or an EPSP. And so way back when I was talking about a depolarizing stimulus arriving at the axon hillock, this is what I was referring to, an excitatory postsynaptic potential, or a depolarization of the cellular membrane which reaches the axon hillock. Sometimes, as shown in this image on the right of the screen, there can be a situation in which one neuron is not sufficient to drive an action potential in another neuron. So these EPSPs, multiple EPSPs from one or more neurons, can be added together, and if the membrane potential happens to reach the threshold, then a new action potential will be initiated in the postsynaptic neuron. Additionally, there are some ionotropic receptors which permit negatively charged ions to enter the cell, and this results in an inhibitory postsynaptic potential, or IPSP. So, and when these channels are activated, the cell becomes hyperpolarized and becomes less likely to fire an action potential. These EPSPs and IPSPs are added together at the axon hillock, and the cell either fires an action potential or it does not, starting the cycle all over again in the next cell. The other type of receptors are metabotropic receptors, which are a little bit more complicated, and I'm not going to go into them in a lot of detail, but essentially they exert effects within the cell by activating other molecules or second messenger molecules, which are called G proteins. For that reason, they are called G protein coupled receptors, or GPCRs. There are over 800 GPCRs in the human body. 800 types of GPCR in the human body, and over half of them are devoted to the sense of smell. These G proteins travel, once they're activated, they travel all over the cell, and they can activate a wide variety of functions, including they can open ion channels, in which case they act in a way that's very similar to an ionotropic receptor, or they can activate intracellular messengers, which do a whole bunch of things that I can't get into right now. So once the neurotransmitter has signaled and has bound to the receptor, it unbinds, and it has to be removed from the synaptic cleft. And it can do this in one of three ways. It can either simply diffuse away from the synapse and go elsewhere. It can be degraded by enzymes which exist in the synapse to break it apart and make it inactive, or it can be taken back up into the presynaptic cell to be recycled. Now, as you can imagine, this chemical process is a major point of interest for neuropharmacologists, and indeed, many common toxins and drugs interact with elements of neural communication. So we already discussed how neural conduction can be interrupted, neural conduction being the firing of action potentials, by tetrodotoxin in the case of the pufferfish. But transmitter release can also be affected by a common toxin, botulinum toxin, also known as Botox. Receptors can be directly activated by drugs, so alcohol, nicotine, opioids like heroin or methadone act at the receptors to activate them or to block them. Reuptake can be interrupted by drugs like cocaine and methamphetamine, producing a buildup of the natural neurotransmitter in the synapse because it can't be removed. Or enzymatic degradation could be affected by drugs. So drugs like monoamine oxidase inhibitors, which are old antidepressants, which are still used today, they work by preventing the enzymatic degradation of some neurotransmitters. Or sarin gas, which recently made headlines for being used as a weapon in Syria, has a similar effect. So let's talk about neuropharmacology. Neuropharmacology is the study of chemicals that alter the functioning of the nervous system. It encompasses both pharmacodynamics, or loosely what the drug does to the body, and pharmacokinetics, or loosely what the body does to the drug. So I'm going to, just like with the other aspects of neurobiology that I've discussed today, I'm going to start by introducing some terminology. This graph on the right side of the screen is called a dose-response curve, and it does what it says on the tin. It shows the response to a drug as a percent of the maximal response that a given dose produces. So as we move from left to right on this dose axis, we see that there are increasing responses, or that there's an increasing magnitude of response. However, at a certain point, this is a very classical dose-response curve shape. And we notice that at a certain point, it kind of tapers off. Once you get towards the 100%, the maximal response, it doesn't matter how much more drug you put into the system, you're not going to get any more effect out of the drug. This point, the maximal response that a drug is capable of producing, is referred to as the efficacy of the drug. And it's a measurement of how strong the drug is. Another important concept in pharmacodynamics is how much of a drug you need to do something. So the measurement that we use for this is called the ED50, and it comes from taking the dose that produces half of the maximal effect. And it's ED50, which stands for effective dose at 50%. And the ED50 can be used to generate a measurement of potency, or a measure of the strength of a drug. So a low ED50 means that a low dose was required to induce a half maximal response. That means that the drug is highly potent. If a drug has a very high ED50, it requires a lot of drug to produce a half maximal response, and therefore we say that it has low potency. I'm going to briefly touch on some different types of drugs that we care about in pharmacodynamics. An agonist is a chemical that binds to a receptor and activates it. So most neurotransmitters are receptors, and many drugs are agonists. Most neurotransmitters are agonists, and most drugs that we're concerned with that bind to a receptor and activate it, those are going to be agonists. A partial agonist is similar in that it binds to a receptor and it activates the receptor, but to a lesser extent than the full agonist. So the efficacy of a partial agonist is reduced relative to an agonist, or to a full agonist. And these are useful therapeutically. So for instance, Chantix or varenicline is a good example of a partial agonist, as is buprenorphine, that is used clinically for smoking cessation in the first case and for opioid use cessation in the second case. The third category of drugs is kind of unique and interesting, and it's called an inverse agonist. And this is a chemical that binds to a receptor, but it actually blocks constitutive activity of the receptor. So we actually didn't know about this class of drug until fairly recently, the last 5 to 10 years. Some receptors are actually active even without a ligand bound to them. And they're constantly going, and an agonist at these receptors can increase their activity, but an inverse agonist at this receptor is actually going to lower the activity of this receptor below the baseline. These are really interesting, and I predict are going to be very useful clinically in a few years once we start discovering more of them. An antagonist is a chemical that simply binds to a receptor and inhibits the activity of the agonist. So an antagonist might not inhibit the activity of the constitutive agonist, of the constitutive activity of a receptor. So it's not going to drive the receptor's activity below the baseline, but it will prevent any agonist from increasing that receptor's activity. Indirect agonists are chemicals that increase signaling at a receptor without directly binding to that receptor. So for instance, I mentioned that some drugs interact with transporters that reuptake neurotransmitters after they've been released. So a drug that might block the reuptake of a neurotransmitter could be an indirect agonist, because without directly binding to the receptor, it is increasing signaling at that receptor. Additionally, some drugs can be related to indirect and direct agonists. Some drugs can be competitive, non-competitive, meaning that they compete with other drugs or they don't, based on where they bind to the receptor. So for instance, a competitive antagonist, if you have a competitive antagonist bound to a receptor and you put a lot of agonist onto that same receptor, that agonist can bump the antagonist out and you can overcome the effects of a competitive antagonist. Conversely, if you have a non-competitive antagonist, it doesn't matter how much agonist you put onto the receptor, you will not have an effect because the antagonist is preventing the receptor from being activated elsewhere. So the last concept I'm going to talk about today is pharmacokinetics, or like I said before, what the body does to the drug. There are four major principles of pharmacokinetics, and they are absorption, distribution, metabolism, and excretion. I'm only going to talk about the first three because the last one is pretty self-explanatory. So absorption is the first principle of pharmacokinetics, and this refers to the fact that in order to exert an effect, a drug must be absorbed into the bloodstream and must enter the body somehow. So when we talk about absorption, one of the main things that we talk about is the route of administration of the drug. And the route of administration greatly affects the rate at which a drug can enter the bloodstream. So a drug is going to get into the bloodstream a lot faster if it's absorbed via the lungs than if it's absorbed via the digestive tract. So when we're talking about routes of administration, there is a huge variety of these, but generally, some of the common ones include inhalation, injection, intranasal or snorting, and ingestion or taking the drug orally. There are many others, but these are some of the most common ones. You can see in these graphs on the right, the top graph here shows the amount of, I believe this is cocaine, the amount of cocaine present in the blood of humans that tried the drug, and then the lower graph is those same humans measuring how intoxicated they feel, self-reporting how intoxicated they feel. And so you can see that the intravenous and smoked routes of administration give a much quicker high to the person who takes them compared to, say, the oral route of administration, which has a very long onset and long offset. So this is really important for determining how addictive a drug is going to be. Typically drugs with faster rates of onset are more addictive. Second principle of pharmacokinetics is distribution, which refers to the fact that once a drug has entered the bloodstream, it needs to travel to the site where it will have its effect. As far as neuropharmacology is concerned, the main concern with distribution is the blood-brain barrier, which is a barrier between the bloodstream and the brain, and only some things are allowed to cross the blood-brain barrier. And whether or not a drug can cross the blood-brain barrier is a function of the chemical structure of the drug. So here are two chemical structures for two closely related drugs, morphine and heroin. Actually, heroin doesn't bind to any receptor in the body. Morphine does. Heroin, once it gets into the body, is converted into morphine, and then it binds to receptors. However, heroin, based on its structure, is able to cross the blood-brain barrier much faster than morphine, and therefore it is much more highly addictive because the rate of onset is faster. And once it crosses the blood-brain barrier, it is converted into morphine, and then it has its effect. So distribution is a critically important principle when we're thinking about drugs. The final principle of pharmacokinetics that I'm going to be discussing today is metabolism, which refers to the fact that many drugs can be modified within the body by metabolic enzymes. This is especially important for drugs that are taken orally because many of these drugs are subject to what's called the first pass effect. And this is the fact that some of these drugs can be inactivated or otherwise altered by enzymes in the liver before they even enter the bloodstream. So some drugs are just completely not available if taken orally. They can't make it to the brain and have an effect. Other drugs can actually be administered in an inactive state, then metabolized into an active state. These are some really interesting drugs that are called prodrugs. So for instance, heroin could be considered a prodrug for morphine because it doesn't actually bind to the receptors. It gets into the brain and then is converted into its active state, morphine. So heroin is an example of a prodrug. So I'm going to kind of end here with a case study, another case study of Suboxone. Suboxone is a combination of two drugs, actually. One is buprenorphine, which is a partial agonist at the same receptor as morphine. So it is commonly used in a slow-release sublingual form, sublingual being the route of administration that refers to placing it under the tongue and letting it dissolve. And this is used to prevent relapse in some opioid-dependent patients. However, some opioid-dependent patients, in a bid to get high, they crush the Suboxone, they crush buprenorphine, and they inject it directly into the bloodstream, giving it a rapid onset and a much greater abuse potential than sublingual buprenorphine. So in order to combat this, to reduce diversion, which refers to the unauthorized use of a drug, buprenorphine has been mixed with its antagonist, naloxone, to create Suboxone. Now, naloxone cannot be absorbed into the bloodstream sublingually. And the result of this is that when Suboxone is taken properly underneath the tongue, it dissolves, the naloxone goes straight out of the body, it's not used, but the buprenorphine has its effect, which is reduction of withdrawal symptoms and prevention of relapse in opioid-dependent patients. However, if Suboxone is crushed and injected, the naloxone prevents the person who injected it from getting high by blocking the effect of buprenorphine. So this is a really good example of a time when we've used the principles of pharmacokinetics to our advantage to create a better drug. In conclusion, I hope I've imparted upon you today the idea that the brain is an incredibly complex organ. It is at the root of all thoughts and behaviors, and many of us are interested in treatment for diseases of the mind, and it's important that these treatments be informed by the basic science of the brain. So it was my hope and my goal today to provide you with a foundational knowledge of the structure and function of the brain in order to help bridge the gap between biology and clinical practice. Thank you.
Thank you, Dr. Thompson. That was a great presentation, I must say. And we are receiving comments of the people are appreciating you, and very large amount of information covered in a very short time, and I appreciate the way you presented. Okay, now moving to the next session of this webinar, that is the question answer session. So this is for all the attendees who are attending this webinar. Please use the chat box to send in your questions so that Dr. Thompson can answer those questions. All right, so Doctor, we have a question from Jose. The challenge of treating Alzheimer's, what is the biggest hurdle? Is it the physiology of the brain that prevents finding a treatment?
That's a great question. So Alzheimer's is a very complex disorder, and we actually don't know the neural basis of Alzheimer's. We've found a few clues. So for instance, some genes are associated with a greater possibility of developing Alzheimer's, but scientists are still working on figuring out exactly why Alzheimer's occurs. And I think that the physiology is one of the most important things in figuring out how we can treat diseases like Alzheimer's. So unfortunately, I don't have a good answer for you other than that science is still working on figuring out the molecular and biological physiology of that disease.
All right, thank you so much. Moving on to the next question from Francis. More evidence is tying nutrition with brain and mental health. That is, hippocampus volume increases with healthier diet, depression symptoms decreasing with healthier diet, omega-3 deficiency seen in psychiatric illness, and the gut-brain axis influenced by diet. Can you comment on these associations?
Yeah, absolutely. So nutrition is coming into vogue as one of the most important features of neuroscience. As I mentioned, the brain consumes about 20 to 25% of the body's energy, and that energy is of course coming from nutrition. So there's evidence that some brain healthy diets, so for instance diets rich in omega-3 fatty acids, can improve cognition and can improve the ability to stave off dementias, and some of that is related to the fact that simply having these better foods in your diet can increase the stability of your neurons. The corollary to that is that there are these, like I said, the brain is an incredibly expensive, energetically expensive organ, and there are certain molecules, we call them trophic factors. So brain-derived neurotrophic factor, or BDNF, is an example of that. Neurons constantly need this molecule in order to be active, and in a state of malnutrition or poor nutrition, brain-derived neurotrophic factor and other trophic factors like that are some of the first things that go, and that can lead to neuronal death and decreases in cognitive ability. So nutrition is absolutely a very important thing. The gut-brain axis is very interesting. There's a neurotransmitter called serotonin. You may know this already. There's a neurotransmitter called serotonin, which is very important for regulating mood and for some other features in perception and things like that. And about 90% of the body's serotonin is actually in the gut. So the gut-brain axis, as we call the communication between the gut and the brain, is critically important and can have a major effect on cognition.
All right, thank you so much. This is for all the people who are attending this webinar. Please send in your questions so that Dr. Thompson will be can answer them. All right, while Dr. Thompson, while we wait for the questions from the attendees, I'd like to share some information with the audience. All right, so this is for everyone who is attending. We are coming up with one more interesting webinar next month as well. The name of the webinar, the topic of the webinar is the 4 phases of anger, model and method for disarming aggression and conflicts. So I'll be sharing the link to, the registration is open for this webinar. I'll be sharing the link using the chat box. And we'll be shortly after this webinar, in few days, we'll be sending you the invitation email so that you can register for the webinar. So I just want you to join us for the next month as well. All right, so I think keeping the time frame in mind, we need to end this session right here. So thank you so much, Dr. Thompson. That was a great presentation and nice answers. And,
thank you.
Yeah, and thank you, audience, for joining us today. If you have any further questions related to webinar, you can send in your queries at webinar@sovhealth.com, or you can use my email address as well. It is s.vashisht@sovhealth.com. Once you leave today's webinar, you will receive a follow-up email within 48 hours after the webinar finishes. In that email, you will receive all the information pertaining to this webinar and instructions to download your CE certificate. And in that email, we'll also send you the link of our website called www.sovinstitute.com, where you can watch this presentation again and download your CE certificate. Now I'll tell you how you can download your CE certificate, because lots of our attendees are facing difficulties downloading their CE certificate. So first of all, you need to visit our website www.sovinstitute.com. So this is the home page for the website, where you can see a login window here. Here you can enter your login credential and just sign up. And if you are visiting this website very first time, you just need to press the register button, and then you can enter your login details, and then you can sign up. After this, you will land up on the next page. Here you can see lots of presentations, so you want to click on the presentation you want to download your CE certificate for. After clicking the presentation, you will land on the next page. Yeah, on this page you will receive all the information pertaining to that webinar and a button at the very bottom. Click to continue to video. You just need to press this button, and all of the people who are attending this webinar, they don't have to watch the video again. They just can skip the video and they can click on continue to quiz button. After pressing continue to quiz button, they will land on the next page where they will find the quiz questions and a survey. They can here you can just have to answer the quiz questions and download your CS certificate. And again, if you have any further questions or confusions, you can send in your questions at webinar@sawwell.com. Thank you so much for joining us today. We hope you found this information valuable. We look forward to bring more interesting webinars each month for you. Keep joining us and goodbye.